Today we will be discussing about intervertebral disc. So as its name suggests, it's a disc-like structure which is present in between our vertebral bodies. So we can also consider it as a jelly-like structure or a cushion-like structure which act to absorb the loading forces or to reduce the weight bearing forces in each vertebral bodies. So its primary functions. What are the functions of this intervertebral disc? It is mainly to separate two vertebral bodies and thereby increasing available range of motion. So rather than a rigid structure or rather than a rigid spinal column, because of this intervertebral disc, it will give you a multiple segments in the vertebral body. So in each segments, we have its own amount of mobility. So because of this intervertebral disc, it will increase the motion in the whole spine or in the spinal column, or it also helps to transmit load from one vertebral body to the next. So because of this intervertebral disc, it will be able to transmit the weight from or the load from one vertebral body to the other, and it also helpful to reduce the compressive forces in each vertebral body vertebral body surfaces. The intervertebral disc, which is make up of 20 to 33 percentage of the length of the vertebral column. So we have almost about 33 vertebra and a 23 intervertebral disc. So this 23 intervertebral disc will compose of about 20 to 33 percentage of total vertebral body length. And when we talk about the thickness of intervertebral disc, it is not actually uniform in all the parts of vertebral column. So it varies from each segment to other, mainly based on the amount of weight it transmits. So when we look into the cervical region, because it's the beginning of cervical column and also a less amount of force which needs to be transmitted from one body to other, the approximate thickness of intervertebral disc in the cervical region is almost about 3 millimeter, and when it comes to the lumbar region, the thickness increases as only because the amount of load transmitted from one vertebral body to the other in the lumbar region is high when it compared with the cervical region. So because of that, the thickness in the lumbar region is also increased to approximately about 9 millimeter. So the thickness of intervertebral disc is mainly related to the amount of force which is transmitted from one vertebral body to another. So if you look into the cervical region, because it's a small area and because there is only less amount of loads which needs to be transmitted from one body to other, the thickness is less as compared to the lumbar region where the higher amount of loads needs to be transmitted. So even though if you look the intervertebral body as a single structure, it is composed of three different parts. First one is the nucleus pulposus, second one is annulus fibrous, third one is the vertebral end plate. So as you can see in this picture, the nucleus pulposus is a is a structure which is situated to the center of the intervertebral disc. So this nuclear pulposus is much more like a jelly structure or with a higher water content structure. And this nuclear pulposus is surrounded by the annulus fibrosis. So annular fibrosis is comparatively a rigid structure when you compare with the nucleus pulposus. So the function of annular fibrosis is mainly to hold the nucleus pulposus in position and also give a structure form for the nucleus pulposus. Third one is the vertebral end plate. So vertebral end plate is much more of a rigid structure when we compare it with the annulus fibrous and the nucleus pulposus. So the main function of this vertebral end plate is to have a good attachment with the adjacent vertebral bodies. So these are the three main components which is composing the intervertebral disc. So all these three structures are composed of water, collagens proteoglycans. So these are the main components in each of these structures in the intervertebral disc, but the amount of each of these components is varies in each of these structures. So the in the nucleus pulposus there will be a high amount of water content and there will be a high amount of proteoglycans as compared to the when we compare with the annulus fibrous and the vertebral end plates. And when it comes to annulus fibrosis, there will be a high amount of collagen concentrations when it compares to the nucleus pulposus. So each one of these structures got its own composition of water, collagen, and proteoglycans, and its varies depends on its function. So fluid and the proteoglycans con- concentrations are highest in the nucleus and lowest in the outer fibrosis and the vertebral end plate. So basically, the nucleus pulposus is a water-like structure. The content of water or the fluid and the proteoglycans is higher in the nucleus pulposus region, but it is much more relatively less in this annulus fibrosis and the vertebral end plate. So as we said earlier, this annular fibrosis and the vertebral end plate is a kind of rigid structure. So there is no high amount of water content in the annulus fibrosis and the vertebral end plate. But when we look into the nucleus pulposus, just because it's a water-like or a jelly-like structure, the fluid content and the proteoglycans are much more higher in the nucleus pulposus, and the collagen concentrations are highest in the vertebral end plate and the outer annulus fibrosis when we compare with the nucleus pulposus. So as we said, the annulus fibrosis and the vertebral end plates are much more like a rigid structure. So there is a high amount of collagen fiber in the vertebral end plate, and there is a higher amount of collagen fibers in the annulus fibrosis, not as much as in the vertebral end plate, but the collagen content is very less in the nucleus pulposus. So it's in a reverse order. So if there is a jelly-like structure that we call as a nucleus pulposus, there is a high amount of fluid and the proteoglycans, and there is a very limited amount of collagen fibers. But when we go to the rigid structures as like the annulus fibrosis and the vertebral end plate, there will be a high amount of collagen and a less amount of fluid or the proteoglycans content. So now we need to look into detail about what are the composition of each of these structures. So it's a nucleus pulposus. Nucleus pulposus is about 70 to 90% of water content depending on the age and the time of day. Basically, in our nuclear pulposus there will be almost about 70 to 90% of water content. So it mainly depends on age and the time of day. So as we grow older, the water content in the nuclear pulposus can come down. So in an aged person or in a geriatric population, the water content in the nucleus pulposus is much lesser as we compare with a young adult. Collagen fibers contribute almost about 50 to 20 percentage of the dry weight, and the remainder of dry weight contains many cells including elastin, proteins, proteolytic enzymes, and chondrocytes, and other types of collagen. So basically, other as we said, 70 to 90 percentage is made up of water content or a fluid-like structure or with the proteoglycans, and rest 15 to 20 percentage is made up of the fibers, collagen fibers, or the protein, all those enzymes, chondrocyte, all those things will contribute almost about 15 to 20 percentage of the content in the nucleus pulposus. The nucleus pulposus has both type 1 and type 2 collagen, but type 2 predominates because of its ability to resist compressive loads. So in nucleus pulposus, there is two types of collagen. Both type 1 and type 2 are present in nucleus pulposus, but the type 2 collagen fibers are much higher than the type 1 collagen fibers, mainly because its ability to resist the compressive forces. The type 2 fibers are mainly designed to resist the compressive forces. Because of this particular function in nucleus pulposus, the type 2 fibers, the type 2 collagen fibers are much more than type 1 collagen fibers. In fact, a very little amount of type 1 collagen is present in the central portion of nuclear pulposus. So there is both the type of collagen fibers, but only the amount of collagen fibers are varies. So there will be a prominent type 2 fibers, mainly to reduce the compressive force or the resist compressive forces, and there will be a type 1 collagen fiber also. So we can compare this nucleus pulposus with a water balloon. So what it basically does, when we compress, it deforms to increase its surface area, and when we keep pressure, it will stretch its surface, stretch its boundaries to increase the surface area and to reduce this compressive forces. So we can compare this nucleus pulposus as a water filled balloon. So whenever we apply some forces to this water filled balloon, the walls or the boundaries of balloon will increase, and it will increase the surface area, and it will reduce this compressive forces. So that's the actual function of this nucleus pulposus. So next is annulus fiber. Annulus fibrosis. In general, annulus fibrosis is 60% to 70% of water, depending on age and the time of day. As we said in nuclear pulposus has almost about 70 to 90% of water content. But when it comes to annulus fibrosis, the water content is actually reducing. It only got about 60 to 70% of water content. And it also depends on the age and the time of day. So age, as we said in nucleus pulposus, as a person ages, the water content in the annulus fibrosis also reduces. Collagen fibers makes 50 to 60% of dry weight, with proteoglycans contributing only to 20% of this dry weight. So as we said, in nucleus pulposus, the amount of collagen fibers are much less than we compared with the annulus fibrosis. So when it comes to the annulus fibrosis, the amount of collagen increases. So in annulus fibrosis, almost 50 to 60% of dry weight is composed of annulus fibrosis, and there is a reduction in proteoglycans. So only about 20% of dry weight is composed of proteoglycans. The remainder of dry weight is made up of approximately 10% of elastin and other cells such as fibroblast and chondroblast. So all other cells will almost contribute about 10%. All the elastin, chondrocytes, all this will have a limited amount of weight which is contributed to the annulus fibrosis. Again, type 1 and type 2 collagens are present. However, type 1 predominates in the annulus fibrosis, particularly in the outer portions. So as we said in nuclear pulposus is much more predominantly with the type 2 collagen fibers. But when it comes to the annulus fibrosis, the type 1 collagen fibers predominates other than the type 2 collagen fibers. The annulus fibers are attached to the cartilaginous end plate on the inferior and the superior vertebral plates of adjacent vertebrae, and to the epiphyseal ring region of the Sharpie fiber. So first we said the nucleus pulposus, which is a central structure. This central structure is surrounded by the annulus fibrosis. So this annular fibrosis attached to each vertebrae by the help of vertebral end body. So there will be a cartilaginous structure or a cartilaginous like structure which is called as vertebral end plates. So this annulus fibrosis will attach to this vertebral end plates or to the adjacent epiphyseal ring region. So next is vertebral end plate. The vertebral end plates are layers of cartilage with 0.6 to 1 millimeter in thickness that covers the region of vertebral bodies encircled by the ring apophysis on both superior and inferior surfaces. They cover the entire nucleus pulposus but not the entire annulus fibrosis. The vertebral end plate is strongly attached to the annular fibrosis and weakly attached to the vertebral body. So it's a much more like a cartilaginous structure, or it got a high amount of cartilaginous portion than the water-like contents. So basically its function is to attach with the vertebral bodies. So the annulus fibrosus will attach to the vertebral end plates, and the vertebral end plates is the one which is going to attach with the surrounding vertebral bodies in superiorly and inferiorly. The vertebral end plates is consist of proteoglycans, collagen and water as in the rest of the disc. So as we can see in other structures of the intervertebral disc like the nucleus pulposus and the annulus fibrosus, the vertebral end plates is also composed of proteoglycans, collagen and water and all other contents of the intervertebral disc. But the only difference is that the composition of the vertebral end plate is different from the annulus fibrosus and also from the nucleus pulposus. In addition, there are cartilage cells aligned along with the collagen. The cartilage of the vertebral end plate is both hyaline and the fibrocartilage. Hyaline cartilage is present closest to the vertebral body and is found mainly in young disc. Fibrocartilage is present closest to the nucleus pulposus. So that are the that are the composition of intervertebral disc. So mainly this intervertebral disc is composed of three structures: nucleus pulposus, annulus fibrosus, and the vertebral end plate. So in between all these structures, the amount of each the composition of each structures is different from one to another. As we see in nucleus pulposus, it's mainly made up of a water-like or a fluid-like structure with a high amount of proteoglycans and with limited amount of collagen fibers. So when it comes to annular fibrosis, the water content actually reduces and there will be an increase of collagen structures. But when it comes to vertebral end plates, there is a high amount of collagenous structure and the cartilaginous structure with a limited amount of water-like structure. So these are this is the main difference between the composition of all these three structures. So next is how this intervertebral disc is innervated and how the intervertebral disc has got its own nutrition. So the intervertebral disc are innervated in the outer one third to one half of the fibers of annulus fibrosus. So in the intervertebral disc, either outer one third or one half of the annulus fibers are innervated in the cervical and in the lumbar regions by the branches of vertebral and sinovertebral branches or the sinovertebral nerves. So these are the nerves which is actually innervating this intervertebral disc. The intervertebral disc do not receive blood supply from any major arterial branches. The metaphyseal arteries arises from the dense capillary flexus in the base of the end plate and the subchondral bone deep to the end plates, and small branches from these metaphyseal arteries do supply the outer surface of annulus fibrosus. So this is how the intervertebral disc got its innervation and nutrition.